to come to the next speaker, to welcome the next speaker, Anabel Sorolla, coming from the Harry Perkins Institute of Medical Research at the University of Western Australia. And she will deliver a talk entitled Development of a Targeted Therapy for Triple-Negative Breast Cancer by using docetaxel-loaded polymeric nanoparticles. Thank you very much.
Hello, good morning to everybody. Thank you for giving me the opportunity to speak in this great conference. It's always nice to come to Europe. Although I work in Western Australia, I am originally from Spain. And today I'm going to present to this European conference my research that has been the past few years in the field of nanomedicine. In our laboratory, what we do is to try to develop novel therapies to target difficult-to-treat breast cancers. And for that, we use novel tools. We use, on one hand, targeted nanoparticles. Then we use also, we try to target transcription factors using a peptide approach. And also we do epigenome editing using CRISPR-Cas9 in order to reactivate genes that are dormant in triple-negative breast cancers, or to suppress genes that are overexpressed, that leads to the risk of metastasis or more aggressivity in these cancers. And we do a holistic approach. We interact with different areas in order to, for example, to deliver epigenome editing tools. We use dendrimers of nanoparticles, and also we do the same for delivering interference peptides into the triple-negative breast cancer cells either in vitro or in vivo. Why developing a targeted therapy for triple-negative breast cancers is so necessary? Triple-negative breast cancers is not the most abundant subtype of breast cancer. It's about 15% all of them. But because they lack of the expression of estrogen receptor, progesterone receptor, and HER2, these cancers are very difficult to treat. They don't respond to common treatments used in the clinic, like tamoxifen or in monoclonal antibodies or small inhibitors of anti-HER2. And that's why it's very, very important to try to develop targeted therapies for these cancers. Nowadays, patients affected by these cancers are usually treated with docetaxel, or also there is a nanotherapeutic approach, which is Abraxane, that are paclitaxel nanoparticles in formal liposomes. The thing is that these therapies are totally untargeted. They, as you may know well, they kill normal cells, normal and cancer cells, both together. And in the search of novel treatments for these cancers, in the past we discovered that there is a transcription factor that is highly overexpressed in these cancers, which the name is NGRAIL1. And actually, NGRAIL1 was included in the PAM50 assay, which is a tool used by oncologists to predict response to docetaxel and other chemotherapies. And also, when this transcription factor is present, there is a higher risk of metastasis in these cancers. NGRAIL1 was initially discovered to be present in the brain, concretely in the dopaminergic neurons, what it plays a role in protecting these neurons from apoptotic insults, which is very crucial in our nervous system. Then and also knockout mice for N1, they develop Parkinson's disease consistent with dopaminergic neuron degeneration. Then we thought to target this NGRAIL1 in triple-negative breast cancer and see what happened. The complicated thing is to target transcription factors. Transcription factors are very small proteins. They act in partnership with other transcription factors and with the DNA. They don't have binding pockets. Their interfaces are very large, and also it's very difficult for an inhibitor to bind there. So then we developed a novel strategy for trying to inhibit these transcription factors, that is using interference peptides. These interference peptides, the way they act is that they they have the same homology of one part of the transcription factor that is essential for the binding with the binding partners. So they compete with the native transcription factor and then displace the binding, also with the DNA, and that prevents oncogenic-dependent transcription of this N1. Here we have developed two versions of the N1 iPeps, an active version and a mutant version. They're differing in 2 amino acids in the hexamotif that are essential for this binding. They also have in the N-terminal position a nuclear localization sequence, which will allow the peptide to penetrate into the nucleus and act there. And also, we re-engineer later on these peptides by adding RGD sequences on the C-terminus. RGD sequences that we found they have high affinity for the integrins that are overexpressed in triple-negative breast cancers. First, when we wanted to design the nanoparticle, we wanted to study which was the most appropriate drug to encapsulate. And then we did some synergistic studies in cell viability to see which drugs act synergistically with our N1 iPeps. And as you can see here, we found that docetaxel acts synergistically in reducing cell viability with docetaxel and N1 iPeps, while doxorubicin not. So that's why we decided to continue with the encapsulating docetaxel. And with collaborators at the University of Western Australia, we designed the following nanoparticle. It's a nanoparticle that is polymeric, made of polyglycidyl methacrylate. This nanoparticle has docetaxel inside, is decorated with polyacrylic acid, which confers a negative charge on the surface of the nanoparticle that allows for the anchorage of our N1 iPeps because they are positively charged in the N-terminus. And then we studied the properties of these nanoparticles. As you can see here, this table summarizes the main properties. They have approximate size of 150 nanometers. The zeta potential of the nanoparticles before the decoration with PAA is slightly electropositive or not that electronegative. When we add the PAA, they become electronegative. When we add the peptides, they become slightly positive, which is a positive thing because then we see a greater internalization of our nanoparticles inside the cells, as you can see here in these confocal imagings. And then we wanted to compare how our nanoparticles with clinically available nanoparticles nowadays, like Abraxane. We treated T11 cells, which is a murine triple-negative breast cancer cell line, and we saw that our nanoparticles present a superiority toxicity. And also, we wanted to treat because we wanted to functionalize this N1 iPeps with RGD motifs. We wanted to see the selectivity of these RGDs for our triple-negative breast cancer cells. And when we treated different versions of the N1 RGD iPeps, we saw a decrease on the cell viability in triple-negative breast cancer cells, either in T11 cells or in the SAM159 cells, while healthy cells like the T3Ts or the MCF10A, they were not that affected by these iPeps. Then we wanted to see how these nanoparticles accumulate in vivo. And for that, we first generated tumors in the mice. These mice is an allograft model, so we injected 1/4 of a million of cells into the mice. And then we wait only 4 days, and the tumors were already palpable, and they are about 50 cubic millimeters. And then we injected intravenously different nanoformulations. So we injected either saline or nanoparticles that are untargeted, and nanoparticles that are targeted with our N1 RGD iPep. And together with these nanoparticles, they have linked the rhodamine to allow the tracking in the animal body. As you can see here, there is an increase of the nanoparticle homing into the tumor. And also we saw an increase in the lungs and the spleen, but a decrease in key reticuloendothelial organs like liver and the spleen. And maybe this increase doesn't sound very, much, but I have to say that it's quite relevant because it is like an increase of 2% of the total nanoparticles in the body. And there was a review in 2016 saying that only 0.7% of the nanoparticles injected to animals or to patients reach the tumor site. So that increase then in this case is quite relevant, the one that we observed in our experiments. Then later on, we wanted to see the effect in vivo in reducing tumor growth of our targeted nanoparticles, and we did the following experiment. First, we injected T11 cells into the mice in the flanks. Then these mice have intact immune system. After 4 days, they generated already the tumors, and then we started the treatments intravenously. We treated the animals 5 times at the times indicated here on the lines. And then we collected the tumors at day 14, and we had to sacrifice the mice already at 17 days after T11 cells inoculations because the tumors were already too big to keep them in the animal house. As you can see here, our targeted formulations, the one in red, the animals treated with this formulation showed a decrease in the tumor volumes compared with blank nanoparticles or nanoparticles that are untargeted. When we retrieved the biopsies and we analyzed the biopsies, we saw there was a decrease in Ki-67 staining, which is marker of proliferation used for pathologists in the mice treated with the targeted nanoparticles. And also we saw an increase in apoptotic cell death according to our tunnel assay done in these biopsies. And later we studied how is the toxicity in these animals. First we did a liver function assessment. We didn't see significant toxicity in the liver based in this assay using these indicators of acute liver damage, which is ALT and AST. Later on, we monitored the tumor weight. We didn't see changes in the tumor weight of the animals during the duration of the experiment. Then we analyzed, with the help of different pathologists at UWA, we analyzed the different organs that the nanoparticles have accumulated, and we didn't see changes in the cytoarchitecture of the tumor, and suggesting that there is not much toxicity in these animals. And just as a conclusion, I would like to conclude that we have functionalized nanoparticles with N1 iPeps, and these nanoparticles are better than the naked nanoparticles. These nanoparticles show superior activity in killing triple-negative breast cancer cells than Abraxane, than clinically approved nanoparticles. And the reengineering of these nanoparticles with the C-terminus RGD peptides improves the tumor homing of these nanoparticles. And then nanoparticles targeted with N1 RGD1 reduce in vivo tumor growth, reduced in vivo tumor growth, induce apoptosis, and they didn't show toxicity in mice. And just to finalize, I would like to acknowledge my laboratory that was crucial for undergoing these investigations together with our collaborators and the funding. And also I would like to thank you all for your attention.
So thank you very much. We have now time for a couple of questions.
Thank you very much for the presentation. It was really, really nice. So I have two questions. The first one is, you were doing this combination assays using docetaxel and doxorubicin with the inhibitory peptide. Do you have any clue why it is working with one drug and it's not working with the other one? That's the first question. And then the next question is, in the in vivo assays that you were showing, were you using free docetaxel as a control to see if the nanoparticle was being more active than the free drug?
Yeah, in the in vivo assays, this is a good question. We didn't use free docetaxel for the in vivo experiments. We just focus on the nanoparticles, but I think will be a good thing to compare either with Abraxane or also with free docetaxel to compare if our nanoformulation has a more positive effect, which I think it is, because first it's targeted, and also it has been shown that docetaxel, when it's encapsulated in nanoparticles, protects the drug, and it allows a slower release, and they are more protected in the circulation. The drug is more protected in circulation. And about the first questions about the interaction with docetaxel and N1 iPeps, and why we think that why it's synergistic with docetaxel, we don't really know. We didn't go into detail on characterizing what is docetaxel maybe inhibiting and why that is advantageous for the iPeps. I don't have the answer for this question, sorry.
Thank you very much for the nice presentation and sharing the data. There are a number of concerns about the use of RGD as targeting agent, and several papers show that the particles tend to stick mostly to the vessels rather than progressing inside the tumor. Did you see any evidence if the particles are then taken up by cells, or if you have a release from the vessels in your case?
In our case, what we saw is that these tumors, these triple-negative breast cancer tumors, are highly angiogenic. You can see that they are completely red. And when we treated the mice intravenously with our targeted nanoparticle, we see that the tumor was becoming more like pale, indicating that probably, yes, maybe it's targeting also the vasculature, and that's why the irrigation diminishes. And also, we decided to actually use, for example, this peptide approach instead of an antibody, because we wanted to actually target an intracellular target, a nuclear target in this case, that is N1, no? And we thought that this was the best approach to link these RGD peptides that are more or less small, that will propel the peptide into the nucleus rather than an antibody.
Thank you very much for your talk. Now